one of the directors of the Center for Simulation and Data Science, and it's a long tradition in the CSD and the preceding organizations that every semester we hold the Charlemagne Distinguished Lecture Series, already since 2010, when Peter Lachs was the speaker, and since then we had many famous personalities giving the talks. Karen Wilcox, for example, Ingrid Dobchis, very distinguished people. And today I'm happy that we can continue this. We had a break during Corona years, but already since the early 2024 we resumed this with Alan Edelman and today with Charlie Taylor. The real introduction will be done by the two organizing persons, Anna Rano and Mark Riegler. I just wanted to mention that I met Charlie probably a quarter century ago when I was visiting Stanford, and he was assistant professor there. And we were discussing FSI simulations, and Tom Hughes was also there and bombarding us with ideas. And I just remember one thing about that time that was kind of standing out, that your server at Stanford was called Solve Death. It was a very nice host name and an ambition, I guess, for everyone there. And today I'm really looking forward to the introduction to the company, to HeartFlow, to their activities, to everything that Charlie has to say. But first I turn it over to Anna.
I will only take this. Do you all hear me well here in the room? I think so, yeah. All right, so my name is Anna Ranno, and I'm one of the associate PhD students of IRTG. It is my pleasure to welcome you today to the Charlemagne Distinguished Lecture Series, where we have the privilege to learning from outstanding researchers across various disciplines. Today, we're honored to welcome Dr. Taylor, a pioneer in personalized simulation-based medicine. Dr. Taylor is a co-founder of HeartFlow Inc. and currently serves on its board of directors. Recently, he was appointed chairman of the board at Ebenbild. Ebenbild is a Munich-based startup developing personalized lung models. He's also a part-time professor of biomedical engineering at the Technical University in Eindhoven. And until recently, he was adjunct professor of computational engineering and sciences at the University of Texas at Austin. Dr. Taylor earned his Bachelor of Science and Master of Science in Mechanical Engineering, as well as a Master of Science in Mathematics from Rensselaer Polytechnic Institute. He completed his PhD in Mechanical Engineering at Stanford University, and then became an associate professor in the departments of Bioengineering and Surgery. He then co-founded Cardiovascular Simulation, Inc. in 2007 and HeartFlow, Inc. in 2010, where he served as Chief Technology Officer and later as Chief Scientific Officer. Over the course of his career, Dr. Taylor has authored more than 450 peer-reviewed publications and holds over 300 issued US and international patents. He has been a fellow of the American Institute for Medical and Biological Engineering since 2010, and was elected to the National Academy of Engineering in 2024. Dr. Taylor is internationally recognized in the field of personalized medicine, particularly for integrating computational fluid dynamics with medical imaging data. Over the past 30 years, his pioneering work has greatly advanced blood flow modeling for patient-specific simulations to aid diagnosis and treatment of cardiovascular disease. So, on behalf of the IRTG and of all students here, we would like to express our gratitude and appreciation for accepting this invitation. And without any further ado, I would ask you to join me in showing our warm welcome to Dr. Charles Taylor.
The microphone seems like it's working here. Great, thank you. It's really a pleasure to be here. You know, as Marek said, we met at a much earlier point in our careers, had a mutual interest in fluid structure interaction and modeling blood flow, and it's really a privilege to be here to give this lecture. I'm going to talk a little bit about really mathematics, bringing mathematics into the diagnosis and management of heart disease. I'll tell you a little bit about the journey and that I've had starting in an academic environment and ultimately bringing this technology out into clinical practice. We'll answer that. I'm surprised you remembered the name of the computer system, Solve Death. And the reason for that is that we had a National Science Foundation group come in, and I remember one of the reviewers was commenting on the mission statement for the lab or what I was trying to do, and he said, you have to keep it really simple, like solve death. And so me and my students, we all laughed about that. It became a running joke in my lab, and said, okay, we'll name the computer Solve Death. It was, of course, that's a really arrogant thing to expect that you're going to do, but it was a joke behind that computer. Let's get started. I'll start off with the first chapter of the story, and this is really starting from the time that I was a graduate student at Stanford. When I started out working in the field, it was a very serendipitous event that brought me into working in the area of modeling blood flow, and it was in part an encounter with a surgeon, a vascular surgeon who had come to Stanford in 1993. I'd started there a year earlier in the graduate program. I was working on problems focused on modeling airflow over airplanes, sponsored by the U.S. Air Force, and I saw a talk that was entitled Blood Flow in Your Health, given by this fellow, Chris Ahrens, who was a new chief of vascular surgery at Stanford. And he came in after collaborating with Seymour Glagoff at University of Chicago, very famous vascular pathologist, who really focused on, among other things, how the cardiovascular system adapted in response to fluid mechanical conditions, whether pressure in arteries or shear forces acting on the endothelium, and also understanding the vascular biology, how atherosclerosis localized in circulation. And Chris had worked with some engineers at Georgia Tech doing experimental fluid mechanics work, really focused on looking at flow through models, glass models of arteries, to look at patterns of flow stagnation and how that related to patterns of atherosclerosis. This is some of the very early work that Chris did, shown on the left here. Again, published now, over 40 years ago, looking at localization or patterns of flow in the carotid artery and how that relate to localization of atherosclerosis. A fellow, Carl Perchtold, in the early 90s started to develop some of the very first 3D simulations of flow in realistic models. Carl was a mathematician in Graz, where now Gerhard Holzapfel, or spent most of his career, but I think was one of the most important figures in the field. Again, a mathematician working in the field and looking at flow patterns. So this is what the field looked like when I started. The first work that Chris and I did together with Tom Hughes in mechanical engineering, Chris and Tom were my PhD co-advisors, was a different problem, but it was still an idealized problem, looking at flow patterns through the abdominal aorta and how the branches of the abdominal aorta altered flow patterns and how those altered patterns in areas of flow stagnation related to areas of atherosclerosis. So this is, again, some of the very first work that we did together. And of course, there were a lot of simplifications, quite frankly, at the time. Simplifications and mathematics wasn't really there. It wasn't, couldn't be applied to solve the problems that we wanted to solve under realistic conditions. In 1995, again, still while a graduate student, I had the opportunity to collaborate with a radiologist that was doing some of the very first CT angiography work, looking at imaging with CT imaging, the first CT scanners, multi-detector scanners, to look at large structures in the body. And this imaging data, when I saw an image of this, I had this idea that we could extract the information, the geometry from the image data, and do the first patient-specific model of blood flow. So this is the first time this had been done, and it was done in 95 and first published in 96. The idea that this then led to was that just like we could use mathematics and computer simulation methods in engineering design, we could use some of these ideas to build patient-specific models and actually plan treatments simulate alternate treatment strategies for patients with vascular disease. So if we're trying to improve blood flow or flow distribution, why not do this on a computer? Plan a procedure. In this case, it was a vascular surgical repair of a patient with disease in the lower extremities, to think about of the alternate ways of doing surgery, vascular surgery. How would each of these techniques, what improvements would be made to blood flow to the lower extremities? So this idea of going beyond the image data to try to understand what the condition of the patient was, to now predict an outcome for an intervention. This is an idea now, it's 25 years ago that this paper was published on predictive medicine in the Journal of Computer-Aided Surgery, really introducing this idea of using simulation-based techniques in medicine to actually plan procedures, simulate alternate procedures, use this information to help guide the treatment strategies. And I'll come back to this at the end, because now, 25 years later, this is happening, every day in clinical practice. So this, again, the first paper, the idea was use diagnostic data to reconstruct a model, a simulation-based planning software system, and then to allow physicians to design improved patient-specific plans. The idea of predictive medicine. So at Stanford, once I finished my PhD, I joined the faculty actually first in the medical school at Stanford in the Department of Surgery, and then had a joint appointment between surgery, first mechanical engineering, and then I was one of the faculty that started the bioengineering department at Stanford and had a joint appointment at the time that I left. And I was really focused on building some of the foundational technology to better extract geometry from image data, to build more realistic physiologic models. One of the challenges that I saw is with the models of blood flow, that it wasn't possible to model flow under realistic pressure conditions, in part because to model flow in the large arteries, you have to account for the rest of the circulation. You have to account for the pumping action of the heart, but also the resistance of flow or the impedance of the rest of the vascular network. And it just wasn't possible. The mathematical models weren't adequate to be able to solve for those particular problems. One of the colleagues at Stanford, Joe Keller, who's one of the foremost applied mathematicians of the last century, was working on this problem of exterior acoustics in this method he called a Dirichlet to Neumann map. I followed a similar approach to adopt these techniques for doing more realistic flow conditions. Some developed fluid structure interaction techniques as well. And again, the focus was how to bring these techniques into realistic problems, realistic clinical problems. The geometry, we started with extracting center lines, doing segmentation, level set methods along the length of the vessel to create geometric models, and then also some direct 3D image processing techniques. So again, a big part of my lab was focused on how do we efficiently extract geometry from image data. The physiology of the mathematics is a method, this is now one of my most cited papers, this method called the coupled multi-domain method. And the idea is a mathematical construct really based upon this idea of Dirichlet to Neumann maps to be able to model flow in the large vessels coupled to any model of the downstream circulation or the upstream circulation. And again, this is an important approach that was taken because with models of the circulation, we have the challenges is that it's so extensive. We have the microcirculation influences flow in the large arteries, so we have to account for that, but we cannot resolve all of the physics, all the geometry of the entire circulation. We have to use models of different level of fidelity, often reduced order models, and then the challenge is how you couple these models together. And then the coupled momentum method was a model for fluid structure interaction that we developed, and had an advantage of being able to do very large scale problems. And this was actually my last doctoral student at Stanford, who then worked with one of my other doctoral students who became faculty, Alberto Figueroa, and this is work that they published after I had left, but really a kind of a full body model of wave propagation, fluid structure interaction, simulation. And it was possible to do this with this approach. There are some assumptions and simplifications that were introduced, but it was then possible to solve for wave propagation problem. Did a lot of work also in verification with analytical solutions. There's a method to verify a 3D solution, or verify a wave propagation model called Wormersley's elastic tube theory. So we did work to validate, or verify, sorry, our techniques that we were developing, fluid-structure interaction, some methods of method manufactured solutions, some work we did in viscoelasticity, and then also some validation work. First with models, not glass models, but plastic models that we built, that we put into a magnetic resonance imaging machine to do phase contrast velocimetry, to measure velocity fields and patterns, and then we did validate our numerical methods. I told this earlier to a group that when I first started work in collaboration with one of our physicists at Stanford was the fellow who developed phase contrast magnetic resonance imaging, Norbert Pelk, and I went to tell him, said, I want to validate my computational methods against your experimental methods against phase contrast MRI. And he said to me, well, that's funny. I want to validate my phase contrast MRI methods against using your computational results. And it kind of introduces this kind of dilemma that when one is doing both and you really understand the limitations, the challenges of doing experimental measurements, sometimes the assumptions that were required to go from what was measured versus to the velocities that were extracted, and vice versa. But did a range of work, did some animal work, large animal work in validation, experimental surgical models. We had a large animal suite that was next to the MRI machine, and Chris Ahrens would do some procedures, large animal procedures that we could then develop these constructs for modeling blood flow and then measuring it with magnetic resonance imaging. So the last part of this first chapter was putting it together to understand modeling blood flow through the coronary arteries and understand the blood supply muscle of the heart. The challenge of modeling flow through the coronary circulation is there are many, not to mention the fact that the coronary arteries are moving, over the cardiac cycle, but also the time-varying contraction and relaxation of the ventricle is either allowing or impeding flow through the coronary arteries. So the physics is really complicated. Likewise, the coronary arteries come off of the root of the aorta, so they're very influenced by conditions in the systemic circulation, conditions right downstream of the aortic valve. So it's a particular challenge to be able to model pulsatile flow, 3D simulations of flow through the coronary arteries. And we, developed an approach using a reduced order model for the heart, a time-varying elastance model for the heart to basically produce a pressure-volume loop for the contraction of the ventricle, a model, what's called a Windkessel model for the downstream circulation of the systemic circulation, and then a model for the coronary microcirculation with a time-varying change in resistance that was induced by the contraction and relaxation of the ventricle. And then we could produce realistic conditions. First paper on patient-specific modeling of blood flow and pressure in the human coronary arteries. So at that point, it was really interesting because it was possible to be able to do it, to model flow through the coronary arteries, but the question is, what is this useful for? Well, around the same time frame, or over this same time period, there was a group of physicians, physiologists, cardiologists that were really starting to use pressure measurements of flow in the coronary circulation to be able to better understand coronary disease and management of patients with coronary disease. And this is where the story will continue. I just want to acknowledge the doctoral students, the 25 doctoral students and postdocs from my lab over the time that I was there. And these are a few of the review papers that I had written in that time frame. One of the last things we did is we open sourced the software. It's called SimVascular. This is now widely available. I think you see there's over almost 50,000 downloads, so far to this date. A lot of my former students went off and have continued to do this work, continued to leverage and develop this tool. Allison and Sean, Allison's at Stanford, Sean's at Berkeley, and both of those were both postdocs in my lab, and they've continued this effort. I want to take to the next chapter. So again, some of the foundational technology that developed over the time at Stanford, from the time I was a graduate student in 93, 94 until I left in 2010. So I founded HeartFlow in 2010 to really improve the diagnosis of heart disease. And this actually is an actual patient with her physician in Austin, Texas, actually looking at the result of her HeartFlow analysis. So what was the problem that we were trying to solve, first of all? When I started to learn more about invasive physiology measurements and pressure wire measurements, it caused me to take a step back and say, if a patient presents with symptoms of heart disease, how do you know whether they really have coronary artery disease? And is that coronary artery disease, is it a relatively mild form of atherosclerosis that could be managed with lifestyle changes and medication? Or is it more severe that the patient needs to have an interventional procedure, coronary angioplasty and stenting or bypass surgery? The challenge is that the tests that are historically used in the US, the vast majority of tests that are performed, there's not direct information, non-invasive data is provided for the coronary arteries. There are tests like stress perfusion, nuclear testing is very popular in the United States, where you're looking at a perfusion deficit in the myocardium, really an indirect indication of whether the patient has atherosclerosis in their coronary arteries. You look at wall motion abnormalities with a stress echo to, again, try to look at the deformation of the ventricle to see whether that there's an explanation for that because of disease in the coronary arteries or an ECG change. And the challenge is you're not directly visualizing the coronary arteries, so you don't know if a patient that presents with chest symptoms has symptoms because there's some other reason that is causing symptoms that are akin to heart disease, or they have disease, but it's diffuse disease or it's microvascular disease. You don't know whether a patient has a life-threatening condition related to disease in the coronary arteries. A result of this, with this indirect testing approach, is that often if you then try to say, if you take a patient to a catheterization lab and say, do they actually have evidence of disease? More commonly, there was a big study that was done at Duke in the US, and they did an evaluation and looked at patients in the cardiac catheterization lab. And what the conclusion was is that if their downstream, their non-invasive testing beforehand was negative, they were more likely to actually find disease in the cardiac catheterization lab versus whether it was positive. So it was very difficult to try to make conclusions about whether a patient had atherosclerosis in the coronary arteries based upon this. Now, there's a lot of reasons for it. I mean, in fairness, you're measuring different things from a perfusion test or an echo versus what you're measuring if you go take a patient to the cardiac catheterization lab. There's much more to the story, but one of the challenges is that often because of limitations in a non-invasive testing strategy, often patients are taken to the cardiac catheterization lab for an invasive evaluation. And what's typically done is if you do traditionally an angiogram is to see is there an area of anatomic obstruction or narrowing in one of the large coronary arteries. If there's anatomic evidence of disease, it's deemed to be obstructive coronary artery disease, and there's a conclusion that is based upon that. What has evolved over time is a realization that with the anatomic evidence of disease, you might have an area of anatomic narrowing that perhaps is a good target for treatment with a stent, or maybe you have a diffusely diseased vessel that might be better bypassed. But it's also important to understand whether that area of disease is limiting the blood supply to the myocardium and putting the patient at higher risk as a result of that. So it's this kind of dual set of information of anatomic obstructive disease and also physiologic flow limitation, which has been deemed to be most valuable, most helpful. And the technique that is, has become I would say the standard, there are other ways of doing this, is a technique called fractional flow reserve. And this is a technique where there's a drug, typically adenosine, which is used to increase flow through the coronary arteries to vasodilate the microcirculation, have more flow through the coronary arteries, and then the pressure downstream in a vessel is measured relative to the reference aortic pressure. And if this ratio of downstream to reference aortic pressure is less than 0.8, then the test is deemed to be positive. It's greater than 0.8 is deemed to be negative. So this is a pressure measurement that is obtained with pressure with a catheter. More typically, if it's less than 0.75, it's deemed to be clearly ischemic, clearly positive if it's greater than 0.8 negative, and then there's a gray zone between 0.75 and 0.8. So this is an assessment of the functional significance of the coronary artery disease, and again, has become a standard for managing patients based upon this physiology information. And the reason for this is that it has been shown to improve outcomes. This is a large study done in the US with a Veterans Administration population over 18,000 patients with stable coronary disease, and showed that if you guide revascularization treatment decisions based upon this invasive physiology measurement, then you have better outcomes. You have better results over time. And it is true that if you just have anatomic evidence of disease, then often you will over-treat areas of coronary disease versus if you have anatomy and physiology together. And again, this is shown a reduction in myocardial infarction rate. The challenge is that this test is done invasively. It's an invasive diagnostic test, physiology test, requires invasive catheterization to get the anatomy and, of course, an invasive pressure measurement. Only about 40% of patients that go into the cath lab based upon non-invasive testing strategy, and this is data from the US, it's probably pretty similar in most other areas of Europe. Only about 40% of the patients have evidence of an obstructive coronary artery disease. So there's a lot of patients, you image them, and some of them don't have any atherosclerosis. Some of them have atherosclerosis, but it's more mild. We've done studies to look at this question based upon non-invasive testing strategies for symptomatic patients. It's typically the case that about 1/3 of patients have essentially clean coronary arteries, no evidence of atherosclerosis. About 1/3 of patients have atherosclerosis that is evident, but it's not too severe. And then 1/3 of patients have obstructive coronary disease. But again, around 40% of patients enter the cath lab of obstructive disease. And if you then do invasive physiology measurement on them, only about 40% of them have evidence of physiologic compromise, a low fractional flow reserve. So if you do those numbers, 0.4 times 0.4 is 0.16. So it's roughly less than 20% of patients with traditional testing strategies go into the cardiac cath lab, have both anatomic focal stenosis, evidence of disease, and also have physiologic limitations. So the argument is that there's a lot of patients that if you could have a better non-invasive testing strategy, you could find these patients non-invasively, then you could have the right patients go into the cardiac cath lab, those patients who are most likely to have disease that could then be treated in a cardiac catheterization lab. And this is the idea, is that how do we identify this before a cardiac cath? The anatomy data, when I first started out and did the very first analyses of flow through the coronary arteries, it was a lot harder to get good quality image data from a CT, to get the anatomic information. Now it's vastly better. And in the last couple of years, Siemens introduced a technology called photon counting CT that is a spatial resolution that rivals what you can get in invasive angiography, and it's 3D, it's volumetric. It's astounding how good this data is. That is, in my opinion, is absolutely a solved problem to be able to get accurate anatomic representation from CT non-invasively. What about the physiology part? And this is basically what we did at HeartFlow, and this is the technology I spun out of Stanford. I always like this quote, from Einstein. It's really about, obviously, as one thinks about a theory versus an experimental approach, you can read it. But my experience is that often, as a theoretician or mathematician, trying to introduce a new technology, these concepts into a field like medicine, there's, by the way, a lot of mathematics that's going on behind the scenes in imaging technologies. By the way, Nico Peels, the physiologist who developed fractional flow reserve, is a mathematician also, who became a physician afterwards. So there's a lot of mathematics but it's often kind of behind the scenes in medicine. And how do you bring this to the forefront with computational methods? So it really is bridging the gap between anatomy and between function. It's providing an anatomic representation and then also detailed physiology information and flow information. I'll come back to that. However, to implement that, to deliver that into a healthcare system, we had to build an entire infrastructure, a cloud-based infrastructure. So the entire system, we have software that sits in a hospital that takes the data out, pushes it to an application running out in the cloud. Our HeartFlow's application runs in Amazon Web Services. So that was a whole, big production, building a cloud-based application to do these types of techniques. And then extracting physiology data. So I always describe this methods as that there's anatomy, there's physiology, and there's physics. The physics is where the computational fluid dynamics comes in. That's, I always say, is the easy part. I gave a talk in Austin, and St. Tom Hughes was in the audience, and I said, this is the easy part. I saw him wince a little bit because, of course, that was 50 years of his career, developing these methods. It's the easy part now. But for us, because of course we're kind of standing on the shoulders of giants, and but the anatomy is is a big challenge extracting anatomy data. The physiology is kind of where a lot of intuition comes into how to take the available data and build a physiologic representation. The reason the image analysis is hard because the data that you get is often difficult to interpret. To get a resolution of a vessel geometry from images like this are quite difficult. But with HeartFlow, it's used in about over 1,300 hospitals around the world, all over the world. Some are really good, high-quality image data. Some have a photon-counting CT scanner that I was telling you about, but many don't. Many have earlier generation CT. Patients have a lot of disease. There's motion, et cetera. It makes it really difficult. But what has evolved over time, we've been able to process data that, quite frankly, are not possible for a physician, a radiologist, or trained cardiologist to interpret. This is an example of a case that we did. This is from New York. It was a study that we did for patients going directly from CT imaging to bypass surgery. You see the white, and you don't have to be, I would say, a physician to know that what you see there in that image on the left is bad. There's a lot of calcium. And it makes it very difficult to interpret where the boundary of the vessel is. It's very challenging. But this is a patient where the surgeon had that information and operated on the basis of this non-invasive data. It was a trial that was published in European Heart Journal, I think it was a year or two ago, that was showing that you could operate even in these most difficult patients based upon the non-invasive data. How do we do it? So we started out with the image data. We had patients that went from CT that went to the cath lab, and then we used invasive imaging. So we used invasive imaging data to help identify, where the boundary really was, and then we adjusted the parameters of our CT image analysis algorithms to best match the invasive imaging data. So these are I showed these are the first patients. This was back in 2010 or so, where we had cross-sectional area down the length of the vessel from intravascular ultrasound. We had CT, and we adjusted the parameters of our CT image analysis algorithms to best match the invasive data. This is a study that was now just 5 years ago, where we had invasive optical coherence tomography, where we could see exactly where the boundary was. With OCT data, you have very high resolution to be able to look at from inside the vessel where the boundary is, and then we could match that. We could co-register the data to the CT data, overlay that, and then learn how to process the CT data in a way that provided the best match to the invasive gold standard data. So this result was published. And you can see, for instance, in this center, one of these center columns, the white, the images there of the CT. It's very difficult to know where the boundary is. You can see it beautifully with an OCT exam. If you overlay this, then you can learn how to process, to find the boundary of a very diseased, calcified vessel by using this type of information together with AI techniques. So the other part of it, and this is a really important part of the success of this enterprise, and also is a lesson for anybody working in the field, is how do you build a framework to leverage artificial intelligence methods. And we've been using AI techniques, first generation machine learning, ultimately deep learning methods that have been in our cleared product now for about 8 years or so. And the idea is that you have an automated algorithm that then extracts part of the process, and then you have a person that looks at the result, the output of the automated algorithm in a visualization system, a workstation, in the context of the image data. So we have a person that is looking at, on the lower right here, looking at the center lines that are extracted from an automated algorithm and checking, inspecting, correcting that. So those corrections that they make, they create a new corrected annotated data set that could be then fed back into future generations of the algorithms that eventually you get to the point with training on, HeartFlow is a repository of almost 500,000 coronary CT scans now. You can get to the point where the automated algorithms cannot be improved by a person, and then you remove the human editing, from that step of the process. So we went from something that would take us days to do the first analyses that now takes about, less than 10 minutes or so from start to finish to do a full interpretation analysis of even the most difficult, complex cases. The other value, so there's the invasive data, there's the human-in-loop AI, and then the third part of this is that anytime we would produce a result of fractional flow reserve derived from CT, and the physician took the patient to the cardiac catheterization lab and did an invasive measurement. If they saw a difference between what we predicted and what they measured, we were required, from a regulatory perspective, to do a complaint investigation. So every time we did that from the very beginning, when we would have a result, we produce it, the physician would say, that's not what I saw in the cath lab, we would do an investigation and figure out why. So that information coming back enabled us to learn, do a root cause investigation, understand when the model was discordant from the measurements, and then start to drive the complaint rate down. Now, there's no technology in medicine, or probably any other field, that has a false negative, false positive rate of less than 1%. It's not possible. Both are underreported, clearly underreported. But if you look at the trends, then what it shows is that with that feedback over time, you can make improvement to the technology, that you can get to a level of fidelity, a level of result that, quite frankly, can be trusted, can be used in clinical practice. So it's pretty rare, it's pretty uncommon now that we have a major discordant case. It does happen, no technology is perfect, but it becomes increasingly rare. That's the anatomy. The physiology, again, the idea behind the physiology model is how do you find the simplest possible model, but no simpler. Einstein again says that, goal of theory is you try to make these irreducible basic elements as simple as few as possible without surrendering a single datum of experience. That's a challenge in figuring out how to do this, and how do you develop a physiology model, that can do this for the purposes. So I've published a few papers on this. This is, the most recent model. Really, the principles are, we have information from a cardiac CT scan about the demand of coronary blood flow, about how much blood flow goes to the myocardium under baseline conditions, because we can look at the size of the myocardium. There's a relationship between how big the heart is, the myocardial mass, and the total amount of coronary blood flow under baseline conditions. How the vessels are organized is actually a clue. When we look at a pattern of blood vessel branching, it's a clue to how much flow goes to a relative part of the territory versus another. So every patient has a unique structure of their vascular tree. You can use that information about the number and size of branch vessels to deduce how much flow goes to a certain territory of the myocardium, and because there's a relationship between vessel size and the amount of flow. And then we can model the hyperemic response, the pharmacologic response of adenosine, to be able to predict what happens when you go to this maximum level of vasodilation. So anyways, there's more to the story here, but we're trying to model the physiologic principles and actually have some of the same assumptions that are used with invasive fractional flow reserve. And then the last part is the physical laws of fluid dynamics. Again, this is the easy part, solving nonlinear partial differential equations for blood flow. Now we started out with full 3D methods. We tested fluid structure. Interaction methods wasn't necessary for fractional flow reserve. Went to more simplified, rigid wall assumptions and models, went down to 1D wave propagation models, et cetera. So now the calculations take just a few seconds or so to perform. In the planner that people use, it's a fraction of a second on an iPhone or an iPad. So this is the easy part in the end. We've done some work with sensitivity and uncertainty quantification to look at what's important in the model. And I would say, suffice it to say that by far the most important variable affecting the accuracy of fractional flow reserve derived from CT is the degree of luminal narrowing, is this titus stenosis, the geometry along the length of the vessel. The physiologic model, it's less sensitive to that. So if we have a good, accurate anatomic representation as compared to an invasive measure, the physiology prediction is quite accurate against the invasive measure. And then some of the other properties don't matter so much. Turns out viscosity of blood doesn't matter that much. Hematocrit doesn't matter that much for fractional flow reserve because it's a ratio of pressures. And then the data is available for the whole tree. The first question that had to be answered was, is it accurate? That was the first thing that I wanted to know, is that we're making this prediction of this thing that's measured with a catheter, is it accurate? We did actually 3 diagnostic accuracy trials before we got our FDA clearance. We've done a total of 5. This is the 4th along the way, where it was also directly compared against perfusion imaging from SPECT and also from PET, water PET imaging, and then also CT alone to look at diagnostic performance. So for producing fractional flow reserve, it's the most accurate non-invasive way of getting that type of information. It's now been published in over 600 peer-reviewed journals, been used over 500,000 patients. In the US, it's really over 95% of the top 50 heart hospitals. And there's now this number it's actually over 1,300 sites worldwide. In the US, it's also 99% of payer coverage. It's all the private insurance companies, Medicare, and now most of the Medicaid plans cover it, so it's really available throughout. Not yet available in Germany. I know there have been some steps forward in CT, actually getting, the irony of course is this is really where cardiac CT came from. You know, Stefan Achenbach did the most important work in cardiac CT imaging, but I think Germany's been one of the slowest countries to adopt cardiac CT imaging itself, or at least pay for it. But this is in process right now, but hopefully it won't be too much longer. We did a whole series of trials. You know, again, there's been over 100 clinical trials. This is a large randomized controlled trial with 2,000 patients, randomization between standard of care versus a FFRCT guided strategy that was done, showing a reduction in unnecessary procedures, increasing cath lab efficiency. So not only are you taking the patients who don't have disease, they're avoiding a trip to the catheterization lab, you're often finding patients that would be missed with other conventional non-invasive diagnostic tests. This is a great example of a study, and in January there will be 5-year data coming out. This is the data from patients with lower extremity vascular disease. If you have a patient who doesn't, most patients that get cardiac testing are patients that are presented with symptoms. But it turns out that about half of the patients that are dying of heart disease have not reported a symptom before they've had a fatal heart attack. So the challenge is that how do you identify those patients? You know, you could absolutely, take heart disease from the leading cause of death and drop it down a couple of notches if you identified patients early with coronary artery disease, because it could be managed medically at an earlier stage. It could be managed with, interventional procedures or surgery. People don't have, it should be a much more rare occurrence where people have a fatal heart attack. The challenge is that how do you do this? Now, the difference between screening for heart disease versus cancer is something that you can do detection and you won't find it in a lot of patients. But if you take patients over a certain age, there's a very high prevalence of having heart disease, of having coronary artery disease. So if you took a patient, you said a population, we're going to take everybody over the age of 50, everybody's going to get a cardiac CT scan, we're going to do screening. The concern of that approach is you will find disease in so many patients that it was not known that they had disease, and then there's more of an impetus to treat those patients. So there's a concern about healthcare costs or explosion of healthcare costs by screening and identifying these patients. So the way to do it is to find patients that are higher risk, to start with a higher risk cohort of patients that might not have presented with a symptom, but they're sufficiently high risk that you're going to find the disease, and you might find disease where your detection of that disease can change management of those patients. So this is exactly a population of patients. One of the highest risk group of patients that have not presented with symptoms, of heart disease are patients who have proven atherosclerosis in their legs and their lower extremity disease or their carotid artery. So if you have disease in your lower extremities, peripheral artery disease, chronic limb-threatening ischemia, your mortality rate is 10% per year. So at 5 years, a 50% mortality rate. They're not dying of the disease in their legs, they're dying of disease in their coronary arteries. So this was a study to take a group of patients. This was done originally in Riga, Latvia. Now there's a large study that's being done internationally. It's called SCORPAD. But it's patients with peripheral artery disease to randomize them between doing what is done today, which is nothing, after they're treated for their lower extremity disease, versus imaging their coronary arteries. And if you find coronary disease, treating it appropriately. Treating it, if you see left main disease, if you see life-threatening coronary artery disease, you treat it. And the result of that is really profound. It's actually an improvement in survival. To take that 50% mortality rate, or the survival rate here, 53% for the control group, and increase that to 76%. So this is 5-year data. There will be 5-year data coming out from the carotid artery study in January. A similar story. So So the idea of actually diagnosing and identifying disease in this group of patients is demonstrated that you can actually save lives, improve survival. So in the UK, this technology is actually mandated in the UK healthcare system. They had a program called the Innovative Technology Program that HeartFlow participated in. And then after that, it was one of the first 5 technologies to be required to be offered in NHS hospitals. It's called MedTech Funding Mandate. After that, they did a study in the UK. You'll like the name of it. It's called Fish and Chips. They always come up with creative names. It's kind of this British humor for clinical studies. Fish and Chip studies looked at the before versus after the adoption of this technology in their healthcare system, and showed that after 2 years of follow-up, having it available in the hospitals reduced all-cause mortality, reduced also cardiovascular mortality. Hard endpoints, again, and this is just over 90,000 patients that were studied in this, showed a reduction of downstream testing. So all the things that NHS thought would be available, did happen. And so this has been pretty important. In the US, in 2021, they changed the clinical practice guidelines and included, for intermediate high-risk patients, included CT followed by FFRCT for stenosis. So this is in the clinical practice guidelines, or stress testing. And then if the FFRCT test is positive or there's a positive stress test, there's what's called a Class 1B indication going on to invasive coronary angiography. So in the US, if you have to justify taking a patient to the cardiac cath lab to do a procedure, you can justify that based upon a FFRCT or a HeartFlow study. And it's in the FDA, this is the first product of its class to be regulated. It's become a standard. This is an FDA document talking about simulation as a medical device, showing that as an example. There's a study that was done a couple years ago in New England Journal of Medicine, AI, looked at all of the FDA cleared products that are AI related and looked at the clinical adoption based upon US insurance claim data. You see the FFRCT, the HeartFlow analysis had the most claims. It was #1. But what was also surprising is once you got through the first 10 technologies, there's so many products that are cleared, but so few of them are actually getting integrated into the healthcare system or getting clinically adopted. That the #2, the diabetic retinopathy, that's Google's work in looking at, the images of the eye to, so again, it's hard to get, technologies like this adopted and paid for. So for the HeartFlow story, we started with physiology, went to an AI-based method to improve the anatomic interpretation of data. It's called Roadmap. This actually, when it's introduced in a healthcare system, we don't charge for that. But then you can do physiology. There's a quantitative plaque product that is now cleared by the FDA, is also paid for in the US healthcare system now. So this is a big step just this year. So you can do quantitative atherosclerosis assessment in the US healthcare system, and this is a story for cardiovascular business talking about, step closer to Medicare adoption. So this is the story, to be able to, just as you have invasive angiography data for anatomy, you can do this with CT, physiology, also non-invasive, and then also atherosclerosis quantification and a characterization. I want to try to, this is an example of an interface that is used for an interventional cardiologist, for example, where they could look at along the length of the vessel. They see the atherosclerosis down the length of the vessel. Can be used to help guide treatment decisions. And we developed a tool. This is also cleared product. This is the idea of prediction. It's called Planner, where it runs on an iPhone or an iPad, where you can actually plan procedures and predict outcomes of interventions. So the idea is that in this 3D interface, you have an area of stenosis, adjust the length of a treatment zone, remove the area of obstruction, and predict fractional flow reserve afterwards. This was tested in the clinical trials published last year. This was a direct comparison against invasive physiology. This was out of the site in Aalst, Belgium, where Bernard de Bruyne is, one of the fathers of invasive fractional flow reserve. They did a motorized pullback of a pressure wire, did direct comparison with the physiologic predictions at over 97,000 matched locations. You see the level of agreement. This works really well for predicting what would happen if you treat a patient a certain way. Again, that was published. This is a trial that's underway, international trial. It's 1,000 patients that are being randomized between a group where their treatment, their stenting procedures, percutaneous coronary interventions are done based upon conventional invasive imaging in the cath lab versus a CT-guided approach. So randomized controlled trial. It's actually about 3/4 of the way enrolled already. Enrollment will be completed next year. And the idea is that you have all this information available from a CT. You know, physiology and the computing is part of it, but you can provide all this information non-invasively to manage patients with heart disease. And this is the trial that was published, I guess it was just earlier this year, still 2024, right? It's called Fast Track CABG, and it was basically showing that you could use and treat patients, do cardiac surgery based upon these non-invasive strategies. Some remarks here. Again, this is a technology that's gone from the academy to the clinic. It's in clinical practice guidelines. It's actually now in the European clinical practice guidelines. You can use it to explain if a patient has chest pain, to defer cardiac catheterization if it's negative, identify the right patients to be treated if it's positive. You can also predict results of interventions, try to use simulation methods for really predicting, something that you might do and determine what's an optimal strategy for treating a patient. It's a review. It's actually one of the first papers out of my PhD thesis in 1998, and 25 years later, a review article that I just wrote just last year. It was a special issue for Tom Hughes for his 80th birthday. I want to just close a little bit about the future, a few slides if I have a few minutes, I guess. And I would just say, first of all, there's more work to be done for heart disease. Microvascular circulation. This image, CT image, is actually of my own coronary arteries. This is about 12 years ago or so. I learned a lot. I needed more branch vessels, coming off my LAD. I have a big ramus intermedius branch that many people don't have. But I learned more about my circulation. But most of my vessels are not shown there, right? Because most of the vessels, they're below the limits of image resolution. You can't see them. This on the right is from a vascular cast. You can see the level of the small arteries, the arterioles that are available. And actually, most of the circulation is not even shown in that vascular cast. In the capillary network, you don't see the venous circulation. But it's been long recognized that you can have symptoms and also you have poor prognosis, not just based upon disease in the large arteries, but based upon the small vessels. So the question is that can you diagnose this non-invasively? You can do it invasively, and the inventors of fractional floors, or Nico Peels and Bernard de Bruyne, came out with a technique called microvascular resistance reserve. This has become a gold standard for doing physiologic assessment. And the microcirculation, and I'll kind of cut to the chase, but if you combine CT, the physiologic assessment of heart flow, together with perfusion imaging, you can actually separate microvascular and epicardial disease. So this is a study that we had, this is a paper that we had presented, basically coupling a 3D model from CT, a synthetic generated model to fill the myocardium, together with a porous media model of the heart, the muscle, to actually simulate flow all the way from the large arteries down to look at a predicted perfusion deficit. This is compared against a PET perfusion deficit from water PET imaging. So it's possible to go a little further. The other area that I think is really exciting right now is how do you put all this information to predict risk of heart attack? Information about plaque characteristics, geometry narrowing, physiology together. Written a few papers on this idea between plaque strength and stress and vulnerability. This is a paper that's just published this year that put all this together. And the storyline that came out of it is that the pressure difference across the lesion, together with the plaque characteristics, would enable you to predict lesions that then go on to rupture. And this is a study that looked at patients who had a CT scan and later on had a ruptured plaque and presented with acute coronary syndrome. So there's a whole story behind that. It's possible, and I would just say also there's a lot of, I'm going to just close in a couple of slides, but to translate this into clinical medicine, it's been a long journey. It's a long process. I'd say this is what it really looked like. It was not linear. There was a lot of challenges along the way, in translating, the first version of our product that we tried to go through regulatory clearance. We didn't meet our endpoint for the clinical trial. We had to redo the clinical trial. There's a whole process of going through reimbursement. You get regulatory clearance, you don't get paid, It took 4 years after we had regulatory clearance in the US to get reimbursement from Medicare. We still don't have it yet in Germany, for example. So it's a long process to be able to introduce the technology. Even after that, you have prior authorization barriers. This has become really, people have learned a lot about this in the last couple of weeks or so, because in the US, I don't know if you heard about this, but the CEO of United Healthcare, one of the biggest groups, was killed in New York City, and apparently it was related to issues of denial of claims of, in the US it's common that you go in, you try to get a procedure, and then there's claims, your insurance company has to go through a prior authorization process that can oftentimes result in denial of care or a slowdown in the process of getting so it's really hard to get new technologies built into the healthcare system, because there's a lot of barriers to that. And then there was the pandemic, also had a big impact on diagnosis of patients with stable coronary disease, and a lot of other challenges. So not an easy journey, but it is, I would say we did go from 0 to 1. This is the first technology of its class to come out, and I think, there's a lot of opportunities in the future to go from 1 to many. I think there is an opportunity in digital twins or in this type of area, healthcare, to think about a lot of other clinical problems. I'm working with a German company to try to do for the lungs what HeartFlow did for the heart, and I think we'll see a lot of other technologies like this that'll ultimately go forward, and will be translated out of a university environment into clinical practice. So thank you very much for your attention. I'll close there and take any questions that you have.
I think we have time for some questions. I'll come with the mic.
Yeah, thank you so much. That was truly inspirational.
Thank you.
I get a question. I mean, this data that you have, like the patients in the background, they are all like super individual, probably, right?
Yeah.
And so my question would be, in that true positive versus true negative, there's also a third option, which is that the simulation cannot tell you something given the certain anatomy that I am given, right?
Yes.
So how do you handle these situations? Are there also outcomes such as, like, given the specific patient, you have to be careful with handling this
Yeah.
simulation results?
I think that's where it should go. The point that you're making is absolutely correct. But let me take a step back. And sometimes we say, we're being judged based upon an invasive measurement that's done in a cardiac catheterization lab, and that's ground truth. But what we've learned in all of these studies that we've done, one of the studies we've done most recently in the validating this planner, where we did these very detailed measurements, about 30% of the measurements of the pressure traces had to be thrown out because they were deemed by an independent core lab as not being valid. So that's the other part of it that I didn't talk about, that one has to be so extraordinarily careful with the measurements as well when then doing a comparison. So every time I go to a new clinical site, I'll say, okay, well, these are what you have to think about how you acquire your CT data. You have to follow clinical practice guidelines for acquiring the CT. You have to use sublingual nitrates. All of these things that you can't just assume they're going to do when they do the pressure measurements. You want to get the highest fidelity measurement that's possible. But if you do repeat measurements of a pressure wire, sometimes you'll get different results. So you're never going to predict better than the coefficient of variation of your measurement technique. And indeed, there are some cases where, because of the image data, the best that you can do will be an uncertain solution, where it could be positive or negative based upon their confidence interval. I think in the future, what I would like to see is that not just point estimates that are provided, but confidence intervals, to go in and say, okay, here's the result, and here's, for this lesion, this is the confidence interval that we have. And it might be positive or negative. The confidence interval could span that binary cut point. And I think that's the direction to go in. But I've tried to field test that with physicians, and I just get a lot of blank, stares. In some cases, because the idea that our solutions are they're not deterministic, right? It's a stochastic, it's an inherently uncertain problem. But the truth is, every imaging technique they use has uncertainty that's built into the interpretation or the extraction or reconstruction. But it's not But if you do an image like a SPECT image or a PET image, you get one image. You don't get the family of solutions that based upon the uncertainty. So, but you're exactly correct. That's where it should go in the future, where we should be able to say this result, we have much more confidence that it's positive or negative, a high degree of confidence it's positive or negative. This result, because of the nature of disease or the image quality, we have a less confidence in the result. That should be what happens in practice, but we're not there yet for introducing that.
Thank you.
But very, very good question. Yeah.
Hello, Mike Frick. I'm a cardiologist, imaging cardiologist here from the hospital.
Yeah.
I would like to ask, I think it's really good technique. I hope you'll get it soon.
Yeah.
Germany. I would like to ask, especially in complex coronary artery disease, we would like to have information about myocardial viability
Yeah.
sequencing revascularization.
Yes.
Is there any information about myocardial viability?
No direct information. There's some indirect information because I'll give you an example. Is that if we take the epicardial tree and we could have territories of the myocardium that are not viable, and the assumption is that the vessel that is feeding that has adapted to this lack of viability. Because if you have an area of scar tissue, any vessel in the neighborhood that's going to feed that would not be normal caliber vessel. You would not have a normal network of vessels. So, but that's indirect information, you know, to related to viability. There's no direct information. And what we assume in the calculation, as part of the calculation, we take the total myocardium, we have the network of vessels, and then we assume that the flow is uniform, you know, through the myocardium, you know, per se, or that there's a relationship between the amount of tissue and the total amount of flow. How it gets distributed to the myocardium would be different. It would be based upon the network of vessels. But it's not available today. This is why we're trying to combine with perfusion imaging to get to the point where you can actually predict a perfusion deficit in a territory. Now that perfusion deficit could be due to something inherent in the microcirculation, or it could be inherent or related to the feeding vessel, or it could be some combination of the two. So it's not possible to get viability information today, but I think it would be, that's the goal, is to be able to get that with combining it with perfusion imaging. You know, if you do perfusion imaging, for example, and you look at a deficit, you don't know why there's a deficit, right? You don't know whether it's the feeding vessels or whether it's the microcirculation. But the goal is to be able to bridge that gap, but not possible today. Yeah. All we can say, and by the way, this is all that fractional flow reserve says, right? Is that what we can say is that if you have a stenosis in a vessel, in an epicardial vessel, that revascularizing that you could potentially see an improvement in flow, right? Because there's a pressure gradient. You can remove the pressure gradient, but there's not direct information about flow per se, right? Yeah.
I actually have a question about
Okay.
for last in case
Yeah.
someone wants to go first. Right, then I'll go with my question.
Yeah.
I have a question about the last part of
Yeah.
your presentation. So you showed some simulations where you are basically changing the
Geometry.
the geometry,
Yeah.
right? Is it going to be in the near future an option of choosing, for example, the type of procedure to open up again the stenosis, like a stent,
Yeah.
or even a graft if not possible, or a balloon, or is it too much at the moment?
Yeah, so I think with right now, we started with trying to do planning for PCI with the assumption that the model that we have, the freedom that we give for a physician, is to be able to open up that segment of the vessel completely. So we're assuming a very good clinical result where the vessel lumen geometry gets restored, back to its native values. In the trial that we're doing and where we validated that, turns out that that assumption is a pretty reasonable assumption to make, that after PCI that you restore the vessel caliber adequately, that it does not become, there's no segmental pressure loss. we would like to be able to do predicting a result of a bypass, because in the study that we did, this fast track cabbage, we did a study where patients with complex coronary disease, you don't know whether they're best treated with an interventional procedure or surgery. There's the syntax score, a functional syntax score that is available, but in the future, you would like to be able to say, okay, here's a patient with complex coronary disease. Let's look at PCI, what is the level of complexity of PCI you need to do to get a favorable improvement in flow. Let's plan a surgical procedure to do virtual bypass grafting, and then plan the surgical procedure with all the complexity of that, and then say which one is better for restoring blood flow. I mean, that's just one factor that you would use, but that would be the goal ultimately. And we've got the stenting part of it that works pretty well, but not yet the surgery. Yeah, that's a goal, but not available yet.
Just a quick follow-up
Yeah.
question. Do you have, maybe it's a difficult one, do you have a time frame or when you think this will be available?
For that, I don't, it probably won't, all I can say is when it won't be available, probably won't be available in the next 5 years, because I know it's not being worked on right now. So the challenge is that there's so many, when you bring a new technology into clinical practice, there's so many different directions that you can go in, and what's happening within HeartFlow. And I left HeartFlow a year ago. I've been focusing on other things. I'm still the founder. I'm still on the board, but so I'm not directly involved in the research activities anymore. But what I would say is that there's very much more of a focus on how do you take patients and trying to predict patients that have atherosclerosis to predict risk. So more in the direction of combining imaging of atherosclerosis, AI for extracting information about plaque, combining that together with physiology to predict risk of an event. And the reason for that is there's, quite frankly, from a business perspective, there's a lot more patients that fall into the group of, atherosclerosis, less severe, not the most complex cases. So that, it's more or less, I would say, a business decision. But, I absolutely pushed into the direction of the complex patients because those are, there are patients that are very difficult, very complex. And we saw in this trial where it was extremely useful, for a surgeon to have this information available to them before they'd walk into the operating room to have this data. And it was highly valued. There's just a much smaller number of patients like that versus the much larger population of patients that have more mild atherosclerosis where you're trying to figure out how to manage them. There's an unlimited number of things that could be done. I mean, HeartFlow has just scratched the surface of one clinical problem. I mean, it's a big enterprise, it's a big company now, but it's just scratched the surface of one problem. And by the way, in so many other areas of medicine, there's so many other areas where, this combination of imaging, AI, simulation, can really have a, really powerful impact. There's a question.
Yeah, so you repeatedly mentioned, like, okay, if we were to screen like so many patients, we
Yeah.
could probably discover all sorts of things. Right?
Yeah.
So my question would be, do you have a take on, like, could we use this kind of computational introspection? I call it introspection now to redefine what actually is a disease and what is not. I mean,
Yeah.
you touched upon
Well,
that.
it sure would be helpful to have that data available, right? I think the challenge is that we could, from a non-invasive strategy, let me just take a step back. The idea of kind of predicting which patients that will ultimately have atherosclerosis that will have a heart attack, that's the holy grail of, cardiology, has been for a long time. There's been a lot of invasive strategies that have been tried to adopted for that. And I saw a talk, maybe it was about 5 years ago, it was by Gary Mintz, who is really one of the experts in intravascular ultrasound. His conclusion is that you could really say from an invasive perspective with invasive imaging IVUS, with OCT, near-infrared spectroscopy, and the OCT is getting better and better, you really know what are the high-risk lesions. The problem is that your invasive measurement technique, you could not do that in a screening way, right? You wouldn't take every single person who doesn't present with symptoms, bring it to a catheterization lab and do invasive imaging for all those patients. It'd be too expensive, it'd be too much risk, to do that. So the challenge is that how do you, with the available non-invasive data, can you better risk stratify patients? What's the appropriate role? And there's one of the most famous cardiologists, I think the most widely published physician in the world, Eugene Bromweld, said, he described it that your objective is that you start out with risk assessment, a risk scoring strategy. And then non-invasive imaging, the goal of it is to basically better risk stratify patients, to do a non-invasive imaging study that you can say, this patient based upon non-invasive data, yes, they're in the low risk cohort. They could be followed up. They don't need further testing. They could be followed up, later on. Or this is a patient in a very high-risk cohort. Those very high-risk patients need to go on. They're at a higher risk of having a clinical event, having a heart attack in a short period of time. They need more aggressive further evaluation, invasive evaluation, maybe treatment. So that, I think, is the appropriate role, is this sequential strategy. We start with risk score assessment. You go to a non-invasive strategy. Then you go to further invasive evaluation, maybe treatment at that point. And then the question is, what population do you do that on? Ideally, and there's a big move in the US, and now there are clinical trials that are underway, to evaluate this, to say, what if you did, do risk score evaluation? What would be that patient population you would start with? Would you start with patients over 50, over 60 years old? If you started those over 60 or over 70, you'd find a lot of atherosclerosis, but your ability to intervene earlier would be less, right? If you image patients at 50 years of age, with certain risk profile, then you could perhaps do that intelligently. You would find some patients that are honestly ticking time bombs. I've seen a lot of patients like that where we've done imaging studies. Remember, study was one of our first studies done in Saudi, 45-year-old man, they did an imaging study, very tight, left anterior descending disease, lipid-rich plaque. It was a ticking time bomb. And it was intervened upon, in that patient. And I'm convinced that saved that patient's life. But there are those patients, and then there are the other patients with maybe more stable atherosclerosis. They might have more disease, but it's more calcified, it's more stable. So the challenge is how do you introduce that? And how do you manage the fact that you will do a screening test that will identify the majority of patients that you image will have atherosclerosis. But if you can then treat it early and prove that you can manage the patient with medicine early, then you can avoid some of those downstream complications. Ultimately, it should be cost savings. And by the way, as a patient, I've had multiple studies. I did my first heart flow analysis. I did a CT scan for myself. I've done a more recent study. I know what my coronary arteries look like. It's really empowering as a person, right, to know that. Or if you have a loved one, to know, do you have disease that is putting you at high risk and you don't know anything about it? Would you rather that or know that you have something that you can then, that yes, it's concerning, but at least you know it's there. So I think I personally believe very strongly that this should happen. This will happen. There's a study being done in the UK already looking at that right now, looking at and what they showed in the first version of the study is that by imaging patients, by doing a CT imaging, you could reduce myocardial infarction rate by 40% because those patients knew they had disease and their physicians knew they had disease and they could be managed more effectively as a result. It was published in New England Journal of Medicine. There's a big follow-up study that's underway. This is going to happen, but it's just how do we get from where we are today, where we hope, that, we know all the patient's risk factors, but we just hope that you hope you're not one of those patients that's going to be in that group that has very serious atherosclerosis, disease where you're going to have your first presenting event will be a myocardial infarction. If that answers your question, but it's the best I can.
Thank you so much.
Yeah.
Thanks very much for the fantastic talk. You fantastically outlined your method for finding people, for stratifying risk and for stratifying people who have to go further down the avenue, maybe for diagnostic and potential treatment. But do you see a role of your technique also in silico trials? In modifying, maybe we have approved drugs now for atherosclerosis treatment, antilytic drugs, and furthermore. And maybe we have also more complicated lesions that we want to treat in the future, maybe bifurcational treatment lesions or something like that, where you can maybe predict in a certain cluster of patient groups where treatment with stenting may be beneficial, or where you want to go in another direction.
Yeah.
So basically the question is,
Yeah.
do you see a role also for, not for stratifying people who have
Yeah.
who need treatment, but for developing novel treatments?
Yeah. Yeah, yes and yes in both of those. And it's actually underway. There's some clinical trials are underway right now. There's one trial that's being funded by Novartis to look at inclisiran, use of inclisiran in a patient population. And they don't get enrolled into the trial unless they have a negative heart flow study. So they're negative. They're not a patient going to the cath. They should be managed medically. And then we also are doing plaque quantification as well at enrollment and then 2 years after they're put on inclisiran. So that's underway. That's called a Victorian plaque study. And there are multiple other trials. So a couple of trials have been published showing using physiology as an endpoint to look at changes in physiology, changes in FFR along the length of the vessel, and also their trials are underway to look at changes in plaque and atherosclerosis burden with different, novel medical therapies. I think the goal ultimately is to be able to do that with the question of risk. Because if you look at a drug, for instance, that you have a patient, you image them at enrollment, you see their atherosclerosis, they see some combination of lipid-rich or fibrotic or calcified plaque. And you look at that and you say, well, ideally your drug will modify the amount of plaque or won't change the amount of plaque, but it'll change the high-risk plaque characteristics into lower risk phenotype. Which is what has generally been shown, for instance, with statins, in those trials. But the question is, does it matter, right? Because if you look at it overall, yeah, maybe it matters. But what you really want to know is if you have a patient that is going to be managed, can you reduce the risk of having an event, right? Can you reduce the risk of a heart endpoint or plaque rupture? That's why this latest trial I told you about, the EMERALD-2 trial that was just published in JACC Imaging this year, was really important. We have another study that's coming out that is just about to be submitted to JACC, the Journal of American College of Cardiology, for those of you that are not familiar with the cardiology literature, that is looking at the question of time, because this is a nuance to this question. So if you have a patient, you image them, you see the amount of atherosclerosis they have, you say, I've identified these lesions that are high-risk phenotype of lesions. Those lesions will be lesions that are identifiable at CT that are soft plaque, lipid-rich, low attenuation, a low Hounsfield unit in the plaque. There'll be large gradients of pressure loss across those. That's the single best predictor of plaque rupture, and some other characteristics. So you can identify lesions with those characteristics. And then the question is, if you put a patient on a different medication, can you change their risk profile? That's what you really care about, right? Is that, because you could take a group of patients or older age, like that patient I showed you went to surgery, he has so much atherosclerosis. We see patients with calcium scores in the thousands. They have so much atherosclerosis. You're not going to affect that. You're not going to remove it, but can you reduce the risk of an event? Those are, I think, the real questions that you want to get at. Could you change their prognosis, for that patient population? And that's the direction where things are going. The other thing I would just say about this, when you have a database, you have all this information, you can actually start to learn things about patterns of disease that you didn't know were there. I wrote a paper maybe 7 years ago on a class of patients that had small caliber vessels indexed to the myocardial mass. And it turned out that those patients that were much more likely to have physiologic flow limitation with even a milder stenosis than other patients. We just have a sub-study that's coming out in the CABG patient population that shows all those patients going to CABG, they're all phenotypically in that characteristic. They're small caliber vessels indexed to mass, and those are the patients that are going on to CABG studies. So that was particularly useful. So I would say that there's a lot of information that can be leveraged for better therapy, that could be leveraged for a better understanding of the disease processes as well. But you just have to know how to pull it out of the data. Yeah, thanks for the question.
Okay, I think in the interest of time, we have to wrap up the question session.